Hello students, welcome back to a new video of Sunanda's tutorial. In this video, we will discuss about the DNA replication in eukaryotes, and it is the part 2 or class 2 of molecular biology chapter. Previously, in my part 1 video of this molecular biology chapter, I have discussed about the DNA replication in prokaryotes. Okay, but today we will discuss the replication process in eukaryotes. So basically, the eukaryotic replication occur in 5' to 3' end as in prokaryote, but it adds 3000 base pairs. The eukaryotic DNA replication or DNA polymerase in eukaryotes adds 3000 base pairs per minute. The direction of replication is same in case of prokaryote and eukaryote, but the process is very complex in case of eukaryotes because prokaryotic DNA is naked because they lack histone protein, and eukaryotic DNA in contrast have histone proteins. Okay, so the process of DNA replication in eukaryotes is little bit complex in case of eukaryotes than your prokaryotes where it is simple. Then this replication process where it occur during the S phase of your interphase of cell cycle. As you know, the cell cycle consists of an interphase where the cell prepares itself for cell division, and M phase where actual cell division occurs. So the interphase again divided into three phases. One is G1 phase, then S phase or synthesis phase, then G2 phase. G1 phase prepares proteins which will require during the S phase or division phase or DNA replication phase or synthesis phase. Okay. So in this S phase, the DNA replication occurs. Actually, DNA replication occurs. G1 phase is the preparatory phase. S phase is the synthesis phase or DNA replication occur here. Then G2 where cell checks whether it is ready for M phase or not. Then the actual division phase or M phase. Okay. So at first, uh, the process of DNA replication in eukaryotes is divided into three phases or three steps. One is initiation step, second one is elongation step, third one is termination step. So at first, we will study about the initiation. Initiation. So before initiation, a pre-initiation complex is formed, and there are some proteins which are involved in this pre-initiation complex formation. One is DNA polymerase. So we require this DNA polymerase during our initiation and elongation. First one is alpha, which is actually the primase required for addition of primer. Then DNA polymerase beta, DNA polymerase beta or beta polymerase required for the repair of DNA after the DNA replication. Then DNA polymerase gamma, which is required for DNA replication of mitochondrial DNA. Okay. Then then delta DNA polymerase, which is required for lagging strand synthesis. Then epsilon DNA polymerase required for leading strand synthesis. So all these DNA polymerases are required during the DNA replication process in eukaryotes. Then binding protein, one single stranded binding protein is required to stabilize the single strand. Then a clamp loader. Okay. Then origin recognition complex, which will recognize the origin of replication. Then it will add other protein complexes for doing the rest of the process. Then MCM, which is a helicase or mini chromosome maintenance complex. Then two protein complex, one is CDK or cyclin dependent kinase and DDF4 dependent kinase protein. Okay, next. So this CDK concentration actually controls many things. One is when CDK concentration is low. So when the concentration of CDK is low, it will stimulate the formation of this pre-initiation complex. Pre-initiation complex is formed before your initiation start. Okay. So but it inhibits the activation of existing initiation complex. But when the CDK concentration is more or increased, it will stimulate the formation of initiation complex from the pre-initiation complex. But it inhibits the formation of pre-initiation complex. But when CDK concentration is increased, it will stimulate the formation of initiation complex from pre-initiation complex. But when CDK concentration is low, new pre-initiation complex will be formed. Okay. So after that, uh, the origin, the origin recognition complex at first recognizes the origin of replication, okay, and bind to it. And all these process occur in G1 phase before S phase, because cell prepares itself for synthesis of new DNA. So at first, the origin of replication sequence is recognized by this origin recognition complex, and this origin recognition complex first bind to that sequence. Then origin recognition complex will bind another two helicase loader, one is CDC6 and CDT1 helicase loader. Then helicase loader CDC6 and CDT1 will add another protein complex or helicase called as MCM, okay, or mini chromosome maintenance complex. Okay, it is actually the helicase protein. Okay, so basically it is the pre-initiation complex. So in this point, at this point, the cell moves from G1 to S phase. And at that time, the CDK concentration will rise. When the CDK concentration will increase, the cell move from G1 to S phase, and then another protein complex CDC45, then MCM, then genes complex protein loaded. Okay. And at first, when G1, the cell moves from G1 to S, primer is added. Primer is added by DNA polymerase alpha. DNA polymerase alpha adds primer and some nucleotides to the single stranded, because after the addition of this helicase, they become single stranded like this. They gets unwinded. Okay. So after that, the DNA polymerase are recruited for both strands, because in case of prokaryotes, DNA polymerase 3 can polymerize both the strands, but in case of eukaryotes, uh, for lagging strand they have DNA polymerase delta, and for leading strand they have DNA polymerase epsilon. So the DNA polymerization process will occur by the help of DNA polymerase epsilon in leading strand, but in case of lagging strand, the DNA polymerization will be done by this DNA polymerase delta. So after that, the RFC or replication factor C recognizes the distinction site between primer and nucleotides, and then this RFC will load pCNA ring. Actually, when the double-stranded DNA unwind to form single strand before the polymerization or elongation process, primer will be added. Primer will be added to this part, which will be added by this DNA polymerase alpha, then primer. Then after primer, this DNA polymerase alpha can synthesize some nucleotides, about 10 to 15 nucleotides. Okay, 10 to 15 nucleotides. And these nucleotides will be attached or will be formed by this primer. Okay, primer, then RFC then recognizes this site because this RFC will load pCNA ring here, pCNA ring here, because after the formation of primer and some nucleotides, DNA polymerase alpha will be degraded. So this pCNA ring will confirm the degradation of this DNA polymerase alpha. So pCNA ring will bind here, and rest of the process will be carried out by DNA polymerase epsilon in case of leading strand and DNA polymerase delta in case of lagging strand. Okay, so next process is elongation. So in it is the leading strand, and in case of leading strand the polymerization process starts from here. The primer will bind and the process will run in this direction from 5' to 3' direction, and in lagging strand we have to attach many primers, and short DNA sequence will be formed, which are called as Okazaki fragment. So after the DNA polymerase alpha gets dissociated, then the polymerase epsilon and delta will be recruited. It is not alpha, it is delta. It is delta. After the degradation of alpha, DNA polymerase alpha, DNA polymerase delta and epsilon gets recruited there. Okay, because rest of the polymerization process will be carried out by polymerase DNA polymerase delta and DNA polymerase epsilon. Okay, pCNA ring is loaded and it checks whether DNA polymerase alpha got dissociated or not. So after that, the primers will be removed by RNase method or by FAN1 protein. Okay, so after that, primers got removed. Then NAP1 protein bring histone protein into the nucleus, because after the elongation process, uh, the DNA will be wrapped around the histone protein, and CAF1, CAF1 help DNA to wrap around histone. So these two protein, one is NAP1 and CAF1, uh, help the processes which will occur after elongation. After elongation, the histone protein, this NAP1 protein will bring the uh, histone protein into the nucleus, because histone proteins are formed in the cytoplasm and the DNA replication process occur inside the nucleus. So that NAP1 protein will bring this histone protein inside the nucleus, and CAF1 will help it or help the DNA to wrap around the histone. Okay, next the process of termination. So after elongation and wrapping around the histone protein, this is the closing of polymerization process, and generally the termination site is GC rich region, G G G C C C, like this GC region. Then After the termination process, so after the elongation process, one primer, after the primer removal, one gap is formed here and many gaps are formed in lagging strand. One gap is formed here in leading strand and many gaps are formed in lagging strand, because in case of leading strand we have one primer. So after the process of initiation, the RNase will remove that RNA part or primer part. But in case of lagging strand we have many primers, so many gaps are formed in lagging strand. So in lagging strand, multiple primers are removed. Then we have many free 3' ends. So DNA polymerase can fill that gap. DNA polymerase can fill these gaps. But the end will remain as this. This part will remain as it is. Then this. So after the removal of primers, end primers actually, gap is formed at 5' end, which is called as end replication problem. So telomerase enzyme provide RNA template, and on that DNA strand can be extended to avoid this end replication problem. So we have to protect that telomere sequence from degradation. So for protection, one T loop is formed. T loop is formed by the protein shelterin to protect the end or telomere sequence. So this is all about your different steps of DNA replication in eukaryotes. So after this termination, we will have our newly formed strands of DNA. So in this way, DNA replication occur in eukaryotes. Okay. Here I end this lecture video, and I hope you really enjoyed this and will get benefit from it. And if you liked it, please hit the like button. Please share with your other friends and family members. And please subscribe to my channel to get more videos like this. Thank you.